Welcome to MOOC course on Introduction to Proteogenomics. In today's lecture, we will continue with Mr. Abhijit Dikshit, who will talk about the Droplet digital PCR, which will be continuation of his previous lecture, where he started discussing about the basics of this technology. He will talk about the type of samples one can use as the input for the instrument. Instrument can be used for multiplexing as well. Even up to 8 plex assays using single flow cells have been reported. The principal requirement for multiplexing is the significant size difference between the amplicons, at least there should be 30 base pair difference. So, let us now welcome Mr. Abhijit to have an interactive session on digital PCR technology.
Is everybody clear with the technology, how it works? Anybody has any queries? Yeah, sure. The sample does not bind to the droplet, ma'am. Yes, so these are droplets which are made where your oil is making a cover, right, and the sample and the reaction mix is inside those droplets. It is an amalgamation, missile, in case you know what missiles are. That is how it works. Any other queries? Right, ma'am? You would have to do extraction. You would have to do extraction, right? You can have your DNA or RNA from any source, doesn't matter. Right, ma'am? You isolate your single cells by a flow cytometer or any other technology? So, what you can do is, even if you have a clump, even if you have say a pool of cells, right, you are wanting to look at a particular type of amplicon. No, in case of individual cells, then you will have to have a cell to extract a nucleic acid out of it. If you have a pool, how would the system differentiate between what cells the DNA or RNA coming from? Right. 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 Then you separate. So you want to do a separation by this method? No, separation you will have to do by any other method. The PCR can happen here. The PCR and the quantification later part can happen in this. What you are talking of is also a different technology which we have. Right. So, it is a workflow, ma'am, where you isolate your single cells by flow cytometer and then go ahead with this. Right. Right. Correct. What you are talking of, that technology is also there, which is a different technology altogether. I mean, we can have offline discussion. We have another technology which works for that. A single cell isolation and sequencing. Yeah. At a time, you can have two because there are two fluorophores, there are two lasers, you can have two, but there are publications where people have done up to 8 plex assays with a single fluorophore. What you simply need to do is keep the size of amplicons variable, at least a 50 base pair difference between the two amplicons. So, what it would do is, yeah, now this cluster which you see is from one type of amplicon, right. If another amplicon is smaller or bigger than that, that would give a different layer here. You can quantify each of these clusters individually. For example, in 2D you get it here, right. So, you will have another cluster altogether. So, you can have, I mean, there are a lot of publications that people have gone up to 8 plex assays. Sorry, yes ma'am? For a multiplex? At least a 30 to 50 base pair difference. If you have to go more than 2 plex assays. Right. It is not for single cell isolation, it is after you have a single cell isolation, because that amount of nucleic acid coming out of the single cell is going to be very minimalistic. So, the system is capable enough sensitivity wise to capture even that minuscule amount of nucleic acid.
But for that I need a system that will first isolate
a single cell, right.
RNA or DNA from a single cell and put it for a PCR.
Yes.
Which may not be quite possible in all likelihood.
It's not very difficult, ma'am. You can do a single cell isolation by flow cytometer very easily. A flow cytometer can do a single cell isolation very easily. And as I was mentioning, ma'am, we have another system also, it's another technology altogether, where we can do a single cell right from isolation to this to sequencing. That's a parallel technology, different altogether.
Another one point is when you talk about its real time detection, that's not real time again.
It's an endpoint PCR.
It's endpoint.
Yes.
It's not a real time.
Yeah, no, what I meant was this is a real time display how it comes. It's an endpoint PCR. Yeah. Anything else? Any other queries? Yes, ideal limit what we say is from a 60 base pair to a 350 base pair.
So, if you are going to be thinking of multiplexing and you need to think of these amplicons,
Right.
so the window is quite small and you can actually manipulate.
It's actually not, as I told, what is recommended is from 60 base pairs to 350 base pairs, like is for real time PCR as well. But we have got people who have amplified and specifically quantified up to 2 kBa fragments as well. Very easily doable. So what I am talking of are the ideal conditions which come from the literature which is published when it's been manufactured. What people have done, there are more than around 4,000 publications now, if I am not wrong, with this technology. And people have done things which even we couldn't imagine of.
When you are talking about the mutation, if there is a single slip,
Right.
the primers that you are using are specific for this.
Yes.
So that's when you get those.
Absolutely. So the prime requirement or the best amplification results which you will get with this doesn't depend on the quality of your nucleic acid. It depends on the specificity of your primers. If your primers are specific, the data you will get is absolutely specific. The quality of nucleic acid would not hamper your data. Yes, ma'am.
That means it cannot be done for de novo.
No, it can't be. You need to have a specific primer. Yes. You need to have a specific primer for your amplification that you're looking for.
I didn't follow the y-axis, how it shows amplitude.
And this is the fluorescence amplitude. As each of your droplets is emitting a fluorescence, if there is an amplifiable DNA in the droplet, it will give fluorescence, right? And when it emits a fluorescence, it will be plotted here. If it doesn't emit, it is here.
How does that depend on the size of your template?
Right, and the system is optimized in such a way that your specific length of an amplicon, if it emits a fluorescence, it will emit a fluorescence in certain range. So the system takes care of those aspects as well. Yes, because as I told, it's like a flow cytometer. Yes, at least 30 to 50 base pair difference between two amplicons. Anything else technologically, if I can answer someone? Yes, ma'am. The slide before this, ma'am? Yes? Yeah.
We can do just 8 samples in one patient.
One cartridge.
One cartridge.
Right.
Like
So
in general real time PCR we can do 95 or
96, right. So what?
We can do just 8
8 at a time for droplet generation.
for droplet generation.
So this process, this you're talking of, right? This takes 1 minute. Okay, so you transfer your droplets in a PCR plate. You generate next.
We have to transfer also.
Yes, you're transferring these, right? You're generating your droplets, which are here in this top row
After generating the droplet, we need to transfer those droplets
to a PCR plate and then put a PCR.
variation in this step also.
No, that doesn't happen. It's been well optimized for that. The droplets which are formed are very stable.
In fact, like for statistical significance, we need to do three control and 3 treatment. Suppose we are
You don't need that in this. You don't need that for this technology because each of your reaction is what is splitting into 20,000 replicates. So you don't need to have technical or biological replicates like you do for real-time PCR.
in this like each well it is splitting particular sample size into 20,000 droplets.
Yes, sample size, no, each of your reaction.
Like DNA will be divided into 20,000. So how can we?
DNA won't get divided. That your DNA is not getting fragmented,
It's not getting fragmented.
right?
It is getting separate. Like in your video, it was that in a single when they can you show me the first slide?
First slide for this.
First slide.
This one?
which was separated the strands.
It was in the video? Yeah, wait. I won't be able to forward the video yet because I can't see anything here. Yeah, tell me a query, ma'am.
Like in that slide, when there was a single mutation, you have shown that there is a single mutation in the gene, and when we have a DNA sample in a tube, and it's getting separated into different droplets. That was the point.
It's not one fragment, right? There are multiple fragments.
There is a multiple.
Right, right.
In that you have shown that if we have suppose 100 strands, and
Okay.
the mutation is in just one strand, and these 100 strands are getting separated into multiple droplets.
Your DNA is getting fragment. Your DNA is not getting fragmented; it's getting partitioned.
It is getting partitioned.
Correct.
So how can we consider it as a 3 replicate sample? Like it will be considered as single.
No, what is happening is you put your replicates or triplicates to eliminate most of the times to eliminate your pipetting errors. If you go for a real-time PCR, right, you should not have more than 0.5 CT value difference, right? Right. So here it doesn't matter because the data that you get is copies of your DNA per microliter of your sample. Right.
Maybe those fragment is not there in any of the droplets. So you say that the 20,000 is become 33%. If the same mutation is happening, 40 mutation is happening, it will be 0.1%.
Right.
If you're doing here, you're getting the 20,000 droplets, and then you're saying 33% is going to increase. The question is that each droplet, whether this DNA eats, they have it or not have it.
Doesn't matter, sir. Not all droplets will anyways not have. That's okay. We have publications where people have got 0.0001% of the mutant as well. Right. Right. So the data that comes out comes after implying a Poisson's algorithm, which takes care of your statistical significance. From the publication perspective you are asking, there are more than 4,000 publications already with this technology where this direct analysis has been done. Your term of asking is the SD, right? The standard deviation that you would get from each 3 replicates that you get, correct? That's required because you might consider that your handling might be different, right? There will be pipetting errors to take care of those things. Right, right. Correct. So, but the technology is made in such a way that transfer of droplets, none of your DNA molecules are left behind. The technology makes sure that all of your reaction mix components are split up and are present in the droplets. None of it is left behind in the cartridge. The system is that rigid. It doesn't leave anything behind in the cartridge. Yes. None of the droplets what we make here, for example, in the first step, yeah, these droplets which are made, right? Yes, yes. Nothing is left behind in your cartridge. All of it is getting transferred. Nothing is there back in the cartridge. The cartridge are not, you cannot reuse the cartridge. No, no. You cannot reuse the cartridge. Yeah. It's an endpoint PCR, sir. So that's what I mentioned. There was an entire slide on that, right? Here. Here. It quantitates it. The data that you get is it will give you how much ever DNA or RNA was present in your sample. It gives you copies of your amplified DNA in each sample in copies per microliter. It's not you don't have to do any manual calculation like you do in real-time PCR. You have a CT value, then you do a FALF method, delta CT, delta CT. You don't have to do anything. The system quantitates and gives you the precise value. Yes, ma'am. Yeah.
Is there any limitation on the amount of DNA that we put in the starting reaction with? That is I'm talking about the template.
The minimum amount of DNA you mean, right? Again, we have got lots of data where people have worked on femtograms of DNA as well. Even a single copy spike, you have a company called as Horizon, which gives you copy number controls, right? It gives you 1 copy, 2 copies, so you can spike your sample and check the copies. The system has been validated through that. The template can be of any size, but your amplicon should not be very long. Ideally, should not be above a KB. Right. So, in case you are using genomic DNA, right, we recommend doing a restriction enzyme digestion. If you are using a genomic DNA, which you feel is going to be very long. Correct. Correct. If it's a cDNA, then doesn't matter, but if it's a genomic DNA, right, we recommend doing a restriction digestion, so that your template is available for the primers to bind and elongate. It will go in either of the droplets. It can be 1 droplet also, it can be 10 also, it can be 100 also, it can be 1000 also, doesn't matter, because it reads each of them individually. So, this thing what you see here, even if you have this 1 droplet positive, everything else is negative, you can be sure that that 1 droplet is because there is some DNA which is gone there, which is amplified, and hence there is a fluorescence. Right, ma'am.
So, what are the chances that a particular droplet, can have more than one?
Yes, yes, yes.
So, what are the maximum number that can be quantified?
Above 5, generally about 10 lakh copies if it's there, then it starts, because what will happen is all of it will be positive, then you will have no negative droplets. All of it will be positive. So, you might not get a very precise amount of DNA. That's about 10 lakh copies if you have that abundant amount of DNA in one sample. If you have per sample, per droplet, because 20,000 into 5 becomes that number. So, what happens is the system, while it gives you the data, it takes into account your forward scatter and side scatter, like it does in a flow cytometer. So, even if there are up to 5 copies in one droplet, it quantifies them as 5 or 4 or 3 or 2 or 1. It does not give you as 1 copy. Maximum is 5 per droplet. That amounts to more than 10 lakh copies in your sample. Anything else? Yeah. Right. So, in that case, you would get multiple clusters here. You won't get a single cluster. If it's amplifying, it's binding somewhere else, right? The size of the amplicon will be variable. It cannot be of same size, right? So, it will give a different cluster here. You won't find such clean clusters. You will have another clear cluster, which can be of a smaller or a bigger size. It's very sensitive for that. Because right. So, maybe in case you know one of your standard, even if you have one standard, for the first time if you are putting a new reaction, you have one sample where you know this is going to work for sure, right? You just put that for once and see which fluorescence amplitude does that come to for once, and you know that value next time. So, if you are getting something variable, that means there is something non-specific also which is amplifying along with your specific thing.
In conclusions, I hope today you have learned how ddPCR can be used for multiplexing. It has very high sensitivity and specificity. ddPCR makes sure that the sample is not lost during the droplet formation, hence increases the efficiency of the process. In the next supplementary lecture, we will talk more about various proteomics applications. Thank you.